Okay, in this chapter, we're going to talk about enzymes and how it kind of relates to metabolism. So, the main idea for everybody is that enzymes control the metabolism of the cell. And if you're an HL student, the metabolic reactions are regulated in response to the cell's needs. And that's a part that you need to know, but the SL students do not. Okay? So, from a TOK standpoint, the first question says development of some techniques benefit particular human populations more than others. For example, the development of lactose-free milk available in Europe and North America would have a greater benefit in Africa and Asia, where lactose intolerance is more prevalent. The development of techniques requires financial investment. To what extent should knowledge be shared with regard to techniques developed in one part of the world that are more applicable in another? So go ahead and write that down where you've been writing your other TOK questions down, and then we'll discuss that in class. Okay? If you're an HL student, here's a TOK question for you. Many metabolic pathways have been described following a series of carefully controlled repeated experiments. To what degree can looking at a component of parts give us knowledge of the whole? So for the TOK students, you guys can go ahead and write your answers to that one in your spot where you've been writing these other ones, and then we'll talk about these in class. And certainly if you're an SL student, you can write an answer down to this and we can kind of share out in our discussion. Okay? All right, so metabolism, as we learned earlier in the course, is that that's the sum component of all the chemical reactions that take place within the cell, within the organism. And it's the way that the cell specifically manages the material and the energy resources that it has, okay? And we've got two types of pathways that we've talked about. We have anabolic and catabolic. In the anabolic, we said are the buildup ones. So you can think just kind of refresh your memory what we said earlier in the course, anabolic, like anabolic steroids, which is what some people take when they're cheating in athletics to get larger and gain an advantage. So the anabolic pathways, these are the build-up components of the metabolic pathway. And then catabolic pathways or catabolism, that's the breakdown. That's kind of like digestion, breaking these things down and making the components of these larger molecules useful for the organism. So in other words, what we've said in the past is that you eat food, catabolic pathways break this food down, and the enzymes help that, and then makes the components or monomers, things like that, available for the cell, and the metabolic portion of the cell uses that for the anabolic processes, which could be build up, repair, those types of things. So that's just kind of a refresher on the anabolic and catabolic pathways. So, for instance, one of the common things that we've talked about is we can take the amino acids that are made available through these catabolic pathways, these digestive types of pathways, and we can build proteins out of them. Okay, catabolic pathways, on the other hand, one of the ways that you can look at this, aside from just breaking down, for instance, proteins like we've been discussing, we can talk about breaking down glucose in kind of harnessing the energy in the bonds of glucose to yield ATP. And the ATP, we said, too, is the energy currency for the cell. That's what the cell uses in order to kind of run its metabolism. Okay, so now, as you probably learned in an earlier course, there's two main types of energy. You have potential energy, which is stored energy, and then you have kinetic energy, which is the energy of motion. And the chemical energy we think of as stored energy, and oftentimes like the position of a rock on top of a hill and it can roll down. But you can think of chemical potential energy as potential energy that's stored in the bonds of the molecule, like the glucose molecule. So when we break that glucose molecule down, we can yield energy, or we're giving off some energy that's stored within those bonds, and we can use that to drive other components of the metabolism. So before we kind of talk further about this, I just want to briefly go over a couple things with thermodynamics. It's nothing that you really need to, have a huge grasp on. It's just some general ideas to kind of help you understand some of the energy transformations that are taking place. So thermodynamics is the study of these energy transformations. And when we speak of the system and the surroundings, when we're looking at an actual chemical reaction, the system is the component that we're studying, and then the surroundings is everything outside of that system. And there's two main types of systems that we have. We have open systems and we have closed systems. The open systems, these are one where the energy can be transferred freely to the surroundings. And that's typical of most types of reactions. And then in a closed system, this is one where we isolate the system from the surroundings. And this is much more difficult to do, but just from the studying standpoint, we have open and closed systems. Now, there's a few different laws which govern these energy transformations, and there's two that we're just going to talk about. The first one is the first law of thermodynamics, which says that the energy can't be created nor destroyed. You can only change its form, and that the energy kind of the amount of energy in the universe is constant. You probably learned about this in your physics class or something like that, but this is a pretty common one. You can't create or destroy energy. You can only change its form. You can't create or destroy matter or mass. You can only change its form. They all kind of go together. The second law of thermodynamics might not be as familiar to you, but this deals with entropy or kind of the disorder and randomness, so to speak, of the universe. And what this says is that the amount of disorder within a universe is always increasing, okay? So now when we're looking at these different forms of energy, the only energy really that's useful from our standpoint when we're talking about these enzymes is really the Gibbs free energy or the free energy within the system, and that's the energy that's available to do work, okay? And then there's two other kind of components or vocabulary terms that you're going to want to just kind of keep in mind. You have enthalpy, which is the heat of the system, not necessarily the temperature or how hot it is, but just the amount of heat, and it's kind of related to the matter and the amount of mass within that system. And then you have entropy, which is the kind of randomness or the disorder within the system. And so the Gibbs free energy equation, this delta G here, and in a minute you'll kind of see why this is important. And again, it's nothing that's super important. It's just kind of building a little bit of schema for you so you can understand some things. So when we're talking about the Gibbs free energy here, we have this equation. And what we're concerned with is delta G and the value of delta G, okay? And what this is related to is the change in the enthalpy minus the temperature times the change in entropy. Okay, so we're concerned with what's basically going on here and how it relates to the change in the Gibbs free energy. And when delta G is negative, we say that we have a spontaneous type of reaction, and we say that energy or heat is going to be given off. And it's important for you to know that spontaneity here, it does not mean that it's going fast. It just means that it can occur spontaneously. It doesn't need any kind of input of energy from the surroundings. Certainly if you give it some, it might go faster, but it's going to occur no matter what, basically, okay? And then when we're looking at the different types of these endergonic and exergonic types of reactions, you might have heard them in your chemistry classes, exothermic and endothermic. They're the same type of a thing. It's just kind of the biological terms here of endergonic. And this is where we have a release of heat or a release of energy. And we say that delta G is negative. And that's one of the important things to keep in mind. Delta G is negative, okay? And we say that the reaction is spontaneous. So as these molecules get broken down, they're going to release energy into the environment. Okay, or the surrounding environment, could be inside the cell, whatever. And these endergonic reactions, these need heat in order to occur. So in other words, delta G here is going to be positive, and we say that these reactions are non-spontaneous. Okay, and in order for this, these reactions really to go, they need an external kind of input of energy. Okay, and some of this, as we go through some of the examples, will hopefully make a little bit more sense to you. So these exergonic reactions, if we're looking at the progress of the reaction right here, okay, at this point, you can see that we have a lot of energy available in the system. So these reactants here have a lot of energy, and then when they proceed through the chemical reaction, you can see the products have much less energy, and some of this energy here is being given off. Okay, this is the free energy that's being released. The opposite is true for these endergonic reactions. In other words, we have some reactants here, but we have to have an external input of energy in order for these products to form. But you can see the amount of energy contained in the products is much greater than it was in these reactants right here. Okay, so there was an external input of energy. So when we look at this, what you can think about this, we could think of as photosynthesis. And we'll talk more about photosynthesis in a later chapter. And what we've got is sunlight energy here, okay, powering the reaction of CO2 in water, say, into glucose. And so this glucose now has a lot more energy than the initial reactants did, and some of that energy has come from the sun. Okay? Now up here we can think of this as cell respiration. And now we're going to take this glucose, C6H12O6, and we're going to turn it into CO2 and water, and then we're going to release some energy, which is going to become ATP. So you can see how these endergonic reaction of photosynthesis is used to create products that's used by cell respiration, and that energy that's contained within these products here of photosynthesis can be liberated during cell respiration and used for the cell to do some work. Okay? So how this relates to cells now is we use these exergonic reactions to power endergonic ones. And ATP is an exergonic type of molecule in that it's a triphosphate, and those three phosphates are high energy bonds, and as they're broken, they give off energy to the surroundings, and that energy can be harnessed by the cell and certain components of the cell in order to do work. So we can see here's our ATP molecule, and these bonds within this ATP right here, these are high energy bonds right here. Okay, and when we break one, we get a phosphate, we get ADP, which is adenosine diphosphate, but then this free energy is now available for the cell in order to do work. Okay, so that's how it's gonna go. And when we look at this now, this reaction here releases about 7.3 kilocalories of energy, and this energy then can be used by the cell to power some reactions in a process which we refer to as coupling. So in other words, these endergonic reactions are coupled to the exergonic reactions, and that one of the exergonic reactions here is the hydrolysis of this ATP. Okay? So one of the examples from the book here is we want to synthesize some glutamine and we're going to use glutamic acid and ammonia. And in order for glutamic acid and ammonia to combine with one another, like you see here, in order for these two molecules to combine, we have to have an input of energy, because we see over here delta G is positive. Okay, so this we're saying then is an endergonic reaction. Okay, and so we know that ATP and the hydrolysis of ATP here, we know that delta G is negative, so this is an exergonic reaction. So we're going to use this exergonic reaction to power this endergonic reaction. Okay, so we have a lot more energy here than is needed here, so we can actually do this 2 times. For every 1 molecule of ATP that gets hydrolyzed, we can form 2 molecules of glutamine from glutamic acid and ammonia. Okay, and so what we're showing you here is that these endergonic reactions are coupled to, with exergonic reactions. Okay, so the energy being released is used to power the ones that need it. All right? So ATP now, we say ATP is what powers these cellular processes, and it's regenerated from these catabolic pathways, these breakdown pathways, and this is the process of digestion, and then your body can use the food, can use the energy containing compounds to make the ATP that's needed to run the cell. Okay, and this is, you can see here, the energy from the catabolism is used to power energy consuming types of pathways. So the digestive processes here are going to release or help us make these compounds that can release energy, and then that's used to generate some ATP, which can then power these endergonic pathways within the body. Okay, so hopefully that made sense for you. As always, if you have any questions, make sure you write them down. We can go over these things in class when we talk about some of the TOK questions that we have and so forth. Okay, so talk to you soon.